All right, welcome to Evolution Podcast 8. In this series, we're going to kind of start by examining how we can actually observe evolution happening in real time, which is not normally something we think about being able to do with evolution. So, the first thing we need to do before we get started with anything else is determine what exactly we mean by evolution, specifically who is able to evolve. In evolution, populations evolve, populations of organisms, groups of organisms. The genetic makeup of a population changes over time. Over time, we might see more dark-haired rabbits than light-haired rabbits, for example. That's what we mean by evolution. Favorable traits that allow for greater fitness, greater adaptiveness in an organism, they become more and more common. What does not evolve are individual organisms. One individual born, let's say we have a brown rabbit born, that from a bunch of white rabbits. That does not mean we have evolved a new species, okay? However, natural selection does act on individuals. Individuals will live or die based on how their characteristics they are born with affect their ability to survive in their environment. So, the differential survival of individuals is the mechanism by which evolutionary change occurs, but it is entire populations that actually evolve, not just one individual in that population. And what's really critical is when this differential survival, if you will, some organisms surviving better more often than others, leads to actual differential reproductive success. That's a really important phrase, differential reproductive success. And what that means is that not everyone is able to leave the same amount of babies. Some leave more babies, some leave less. Some individuals do this better than others. And that is the essence of really natural selection causing evolutionary change, is when some organisms leave more offspring with their improved traits and more adaptive traits than others do. Okay? Now, there are two types of evolution that you need to know what those are. The first is called macroevolution. Macro means big, of course, and what we mean is evolution on the grand scale, large scale, big scale evolution, in which we are talking about what we normally think of when we think of evolution. The actual kinds of species on Earth have changed over time. We have gone from extremely, the most simple bacteria being the first organisms, to some perhaps invertebrates of different sorts, and those changing, and then reptiles, dinosaurs, things like that. We are talking big scale evolution. That's macroevolution. Really not easy to observe given our short lifespans. We can see the evidence that it has occurred, but it's hard to observe it actually happening, as compared to microevolution. Micro, obviously meaning small, and it is change on the small scale, if you will, change in the genetic makeup of a population of organisms from one generation to the next generation. So within our lifetimes, depending on that organism, that could be observed. When we think of the classic peppered moth example, those changes in how often we were seeing the dark peppered moths versus light peppered moths, that could be observed within a few generations, so definitely visible. So peppered moths, frequency, color, form change, that we've talked about that. So this kind of microevolution, this small scale change, really leads to many changes that may accumulate over many, many, many years and thus lead to macroevolution. But these small changes as they're occurring seem very tiny and maybe not terribly important until we combine them together into large scales changed over many, 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 many years. Okay, so micro versus macroevolution. Microevolution leads to macroevolution. Macroevolution really can't be observed directly occurring in most cases as we think of it, because it takes thousands, perhaps millions of years. Microevolution, however, is different, and it can be observed directly occurring. Now, just because we might not be able to directly observe macroevolution because it takes so long doesn't mean we can't see the evidence that it has occurred. We can see that in all the ways that we've already talked about. So if we want to, let's focus in on microevolution because we can observe that occurring, we can measure it. How can we measure evolutionary change? Microevolutionary change is what we measure, and we measure it by looking at how the numbers, very quantitative, of different alleles, so flavors of genes, and genotypes, so like alleles, please remember, is referring to like either A, big A, or little a, whereas a genotype is going to be an individual's genetic makeup, so big A, big A, big A, little a, little a, little a, that kind of comparison. So we will measure the numbers of different alleles and also the numbers of different genotypes in a population and see how they change, how those numbers change over time. So before we get into this, let's get some basic terms down for measuring microevolutionary change. The very first term, very important, is population, because that is the level at which microevolutionary change is happening. So remember from ecology, a population is a group of organisms of the same species, and they occupy a certain area in the environment, and they share a common gene pool. Now, we've just introduced another term, so let's identify that one. A gene pool is the total of all the genes of all the individuals in a population. If you've got a population of deer in your neighborhood, if you took all their genes and dumped them into a bucket, that's their gene pool. Okay? Genetic structure is another term we're going to be using. Basically, the term genetic structure is the term for the allele frequencies and the genotype frequencies of a population. Now, what the heck is that, Coleman? So let's identify these two terms, allele and genotype frequencies. Allele frequency is the number that tells us how often an allele is occurring in a population, in a population's gene pool. How often do we see big A? Okay? How often do we see little a in this population? Now, genotype frequency is basically telling us how often a genotype occurs in a population. If we look at our population of deer, how often are we finding a big A, big A? How often are we finding big A, little a, or little a, whatever trait that might be? And remember, this number, this genotype frequency, because when you're talking about genotype, you're talking about a trait in an organism, not just a single allele. We are actually referring to individual organisms, not just their disembodied alleles at this point. So, in order to measure evolutionary change, then, we assume that if evolution is occurring with some trait, then the number of alleles in that gene pool and the number of genotypes in that population will not be remaining constant over time. If evolution is occurring, there is some kind of change going on in terms of the frequency with which we see big A, or the frequency with which we see big A in the population. So the allele and genotype frequencies will be changing over time. So in order to measure this evolutionary change, we must be able to measure how many alleles and how many genotypes at a given locus, a given position in the genome, are present in a population at a particular point in time today. How many big A alleles are in this population? How many big A, and big A, and little a individuals are in this population today? Okay? And then we need to be able to come back later, next year, whenever, and measure how many alleles at the same locus are present a year from now, or how many genotypes, big A's, are present a year from now in this population. If those numbers are different, if they are different from time A to time B, then we should be able to assume that some sort of microevolutionary change may be taking place in that population. Maybe we're getting fewer big A's and there's more little a's. Maybe there's a selection pressure in the environment that is favoring that little a allele. So being able to determine then if evolution is occurring depends on being able to measure this genetic structure of a population, meaning the frequencies of alleles and the frequencies of genotypes in a population over time from point A to point B. So how do we do that? Okay. There's a couple of guys who gave us an answer about how to do that, and their names are Hardy and Weinberg. And so the next podcast is going to talk about what they've told us and how we can use that information to measure microevolutionary change. Okay, that's it for this podcast. I will see you in the next one. Thank you for watching. Take care.